So that's the one I would want to more pay attention to. And again, there won't be labels on it, and you're kind of, labeling it, like it'll have A, B, C, D, and ask you what is A or, whatnot. So, just understanding, you don't have to know the names of these guys, but understanding where is the flow of electrons, where are the protons, where's the ATP synthase, where's oxygen, the final electron acceptor, where's water made, where is this whole thing happening? It's in the plasma membrane of bacteria, right? And that's what we're focused on rather than the mitochondria of eukaryotes. So I think if you know all those components and you can just kind of look at that diagram, kind of know what's going on, then that's sufficient.
So like out of the last complex is where the oxygen meets with the H and creates H2O?
Right, so oxygen is the final electron acceptor right here. It's receiving out of that last complex, it's receiving those electrons, and then also, as the protons are allowed to pass through the ATP synthase and kind of powering that phosphorylation of ADP, then the protons combine with oxygen and that makes water. If it was anaerobic respiration, you might have like sulfate over here, and then when the protons combine and the electrons, you get something like hydrogen sulfide, H2S. Or if it's nitrate, you'll get, it depends, they can produce ammonia or different types of nitrogen compounds as well.
Are those protons hanging out in the cytoplasm, or are they like attached to these proteins along the membrane? Like how do
The
they?
protons are, so this is in the cytoplasm, and this is exterior, but not completely out in the environment, right? Because this is the plasma membrane, and then of course we have a cell wall. So this is that periplasm, that gap between the plasma membrane and the cell wall. So it can contain those protons and create that gradient. In the cytoplasm, I imagine they're a little more diffuse out, and that's why you do get that gradient, where there's definitely a lot more protons up in that periplasm and less in that vicinity of the cytoplasm. I imagine others, when they come in, besides combining with oxygen and making water, they probably combine with other things and make other molecules as well, because that's what, especially charged molecules like protons are going to be attracted to negatively charged molecules, like maybe chloride ion or whatnot that might be in there, forming different things, sodium, chloride, salt is definitely there in cells, and so, I don't know how much HCl is there, but there might be a little bit, I suppose.
So, sorry. These protons are coming from what then exactly? They're just like in high concentration within the cytoplasm?
Yeah, they're getting dropped off from the electron carriers because, we talk about like oxidation reduction and that it's the loss of electrons, gain of electrons, but it's not just electrons. Protons are always there too.
Okay.
We just kind of ignore them, but when we have oxidation, there's also dehydrogenations, removal of hydrogens as well. That again, we kind of push them to the side and give more attention to the electrons.
Okay.
But the protons are there too, and that's where they're coming from that oxidation of those fuel molecules.
Okay.
Yeah.
So, the NADH plus would be the oxidation, and NADH would be the reduction?
Yes, NAD plus is the oxidized form, and NADH is the reduced form. Yeah, so when they say like no examples on, what number is that? Number 5 on chapter 7, no oxidation reduction, including examples from glycolysis, TCA, ETS. Just be able to recognize things like that, like in glycolysis, what's being oxidized? What do we start with? Glucose, right? That's being oxidized. What's being reduced? Just said it. NAD plus to NADH, right, is being reduced because it's gaining electrons and protons. And when we go into TCA, what's getting oxidized?
pyruvate.
Yeah, or acetyl-CoA, either one. Generally, I would indicate, well, I would say, pyruvate gets converted into acetyl-CoA. But yeah, it's acetyl-CoA that actually enters TCA, but I think it's important to still make sure we know that that came out of glycolysis, that it's like a continuation. So that's getting oxidized again, reduction is occurring to the NAD+, right? It's gonna also, again, receive electrons and protons. ETS, it's in the chain. I mean, this is getting, as it gains electrons, it's getting reduced. Then it gets oxidized when it passes them on. And then this one gets reduced, and then oxidized, reduced, right? And that's why it's a redox chain, it's a chain of redox reactions occurring down that. And that's why some books call it ETC, electron transport chain. Others call it ETS, electron transport system. But it is really a chain where those electrons are being passed down, and those complexes, those molecules are being in turn oxidized and reduced as they pass down electrons. Yeah.
So ATP is just given off as a result as it goes down the chain for number 8? Question
Right,
number 8.
ATP is the, kind of the major reason for all of this, right, is that this whole process, it powers that phosphorylation of ADP to make ATP. Because again, it takes energy to put that phosphate on there. It doesn't want to go on there because of the oxygens on the phosphate group, those negative charges. So all of this is what powers that production of ATP. The electrons being passed down, going to oxygen, which is kind of like a magnet pulling the electrons towards it, and the protons creating that gradient that kind of gives that potential energy, like the hydroelectric power example. That's what's providing that. But yeah, where did all that come from in the first place? Well, that's where we always trace back to, well, where did NADH come from? It came from glycolysis, and TCA from the oxidation of glucose or other fuel molecules as well. Because remember, like if the bacteria were breaking down protein into amino acids, those would feed into the cycles as well, or if they're breaking down fats, those will feed in as well. But of course, glucose is the major carbohydrate, so we do focus on that one. Yeah.
And then do we, for question number 6, do we need to know the specifics, like when glucose molecule goes in, 2 pyruvate come out, nothing like that?
No, so for number 6, know what happens in each, and just in the general sense, what goes in, what comes out, no, you don't need to know any numbers. Though.
Okay.
Yeah. Any other questions on chapter 7?
Yeah. Yeah.
Yeah, so basically, this is the proton motive force, that whole proton gradient situation. So besides powering the production of ATP, that proton gradient, that potential energy, like water behind a dam, can power other things as well. Can power the flagella, it can power the efflux pumps that bacteria have, as well as other pumps that can bring in nutrients, or so the pumps can go both ways, of course, bring things in or pump things out, but efflux specifically are ones that pump out, and we kind of talk about them because I find it crazy that bacteria can actually pump out antibiotics, specifically. It's pretty amazing with those, but of course, again, that requires energy to do that, so that's why they use that proton motive force. What else is powered by the proton motive force in this sense? Flagella, pumps, ATP synthase. I think that's the main ones, right?
What's readily available fuel source, right? It's not stored in, it's just when needed, it can move into the cytoplasm, and that gradient creates energy, right?
Right, I mean, it's pretty much always there. Again, like water behind a dam, I guess, unless there's a complete drought and the, lake dries up, And same with the cell, as long as there's food and they're metabolizing, there'll always be that proton motive force available as long as they're able to carry out glycolysis, TCA, and the ETS.
Can you explain fermentation and how it compares? I understand aerobic and anaerobic, but not so much fermentation.
Yeah, so fermentation, is that number 10?
Yes, number 10.
Yeah, so fermentation, main things to know is that it has glycolysis, it's the same, there's no difference there, glycolysis occurs, but the organism, and a lot of organisms that do fermentation, they're also able to do regular aerobic respiration, and then fermentation is just an option if there's no oxygen available. Or even sometimes they just will do it even when there is oxygen available. But they definitely would prefer to do aerobic respiration first because they get more ATP out of that, like we covered. You know, the oxygen holds those electrons and really drives that whole process. But with fermentation, they can't, if there's no oxygen, they cannot do the TCA. Part of it does require some oxygen. So that's again why also anaerobic respiration yields less ATP. And then they can't do ETS. So what they do is, like I said, that NAD plus has to be regenerated. So if they do glycolysis and they've got all this NADH built up, and they're not going into the ETS to regenerate the NAD plus, you have to regenerate the NAD plus somewhere, or the whole cell will just die and break down because it can't continue metabolism if it can't drop those protons and electrons off somewhere. So the cell takes the protons and electrons off of NADH and actually puts them back on pyruvate, or a derivative of, depending on what they're fermenting, but it's usually pyruvate or pyruvic acid. But they do it in different ways with different enzymes and different pathways, and that's where they yield different end products, possibly, you know, ethanol and CO2, or possibly lactic acid or other types of acids. So just know that, you know, that it involves glycolysis, but then after that, the NAD plus has to be regenerated, and so it has to put them back onto that pyruvate. And so what happens is basically there's an incomplete breakdown of glucose, right? Because whatever that end product is, it could have still theoretically, you know, pyruvate could have still been broken down a lot more. So that's why there's less ATP as well, besides just not doing the ETS and the lack of oxygen. They're not completely oxidizing glucose to its maximum, you know, ability to be oxidized, and therefore they're not getting as much ATP out of that whole process.
So then that means aerobic respiration creates the most energy, right?
Yeah,
ATP.
aerobic is the most efficient, followed by anaerobic. Then fermentation is very last in the production of ATP.
Okay, thank you.
Specific fermentation products?
No, I know I mentioned during lecture maybe, because in the past I've asked questions about, you know, recognizing some of the fermentation products, but I did not this time, so you don't have to know that. We'll definitely look at fermentation more in lab when we start our unknowns very soon, so you'll be doing some fermentation tests. Chapter 7 is the toughest, huh?
That's right now.
Yeah, we're ready for chapter 8?